Hello students. Today we will be learning about the part 4 of chemical thermodynamics, in which we will be dealing with redox reactions and redox potential. In many chemical reactions, electrons are transferred from one atom or molecule to another, and the subsequent release of energy that can be coupled to other reactions. These reactions involve the loss of electrons by one atom or molecule, which is called oxidation, and the gain of electrons by another atom or molecule, which is called reduction. The flow of electrons in oxidation-reduction reaction is responsible directly or indirectly for all work done by living organisms. Such reactions in which one molecule is reduced and another is oxidized often are referred to as redox reactions. In non-photosynthetic organisms, the sources of electrons are reduced compounds, such as the food that we take in. In photosynthetic organisms, the initial electron donor is a chemical species excited by the absorption of light. The path of electron flow in metabolism is complex. Electrons move from various metabolic intermediates to specialized electron carriers in enzyme-catalyzed reactions. The carriers in turn donate electrons to acceptors with higher electron affinities with the release of energy. In this lecture, we will discuss the general types of metabolic reactions in which electrons are transferred. We will also learn about how energy changes are measured in such reactions in terms of electromotive force and the relationship of this EMF in volts as reduction potential with free energy change, which is in joules. Cells contain a variety of molecular energy transducers which convert the energy of electron flow into useful work. Now let's see how the flow of electrons can do biological work. Every time we use a motor, an electric light or heater, or a spark to ignite fuel in a car engine, we use the flow of electrons to accomplish work. In the circuit that powers the motor, the source of electrons can be a battery containing two chemical species that differ in affinity for electrons. Electrical wires provide a pathway for electron flow from the chemical species at one pole of the battery through the motor to the chemical species at the other pole of the battery. Because the two chemical species differ in their affinity for electrons, electrons flow spontaneously through the circuit driven by a force proportional to the difference in electron affinity. That is called the electromotive force. The electromotive force can accomplish work if an appropriate energy transducer, and in this case a motor, is placed in the circuit. The motor can be coupled to a variety of mechanical devices to accomplish useful work. Living cells have an analogous biological circuit with a relatively reduced compound such as glucose as a source of electrons. As glucose is enzymatically oxidized, the released electrons flow spontaneously through a series of electron carrier intermediates to another chemical species, such as oxygen. The electron flow is exergonic because oxygen has a higher affinity for electrons than do the electron carrier intermediates. The resulting electromotive force provides energy to a variety of molecular energy transducers like enzymes and other proteins that do biological work. In the mitochondrion, for example, membrane-bound enzymes couple electron flow to the production of a transmembrane pH difference, accomplishing osmotic and electrical work. The proton gradient thus formed has potential energy, sometimes called the proton motive force by analogy with the electromotive force. Another enzyme, which is the ATP synthase in the inner mitochondrial membrane, uses the proton motive force to do chemical work. That is, synthesis of ATP from ADP and inorganic phosphate as protons flow spontaneously across the membrane. Similarly, membrane-localized enzymes in E. coli convert electromotive force to proton motive force, which is then used to power flagellar motion. The principles of electrochemistry that govern energy changes in the macroscopic circuit with the motor and battery apply with equal validity to the molecular processes accompanying electron flow in living cells. Oxidation reductions can be described as half reactions. Although oxidation and reduction must occur together, it is convenient when describing electron transfers to consider the two halves of an oxidation-reduction reaction separately. For example, the oxidation of ferrous ion by cupric ion. You have Fe2 plus Cu2 plus, which gives Fe3 plus Cu plus. This can be described in terms of two half reactions. The ferrous ion is converted to a ferric ion plus 1 electron. And second, the cupric ion is converted to a cuprous ion while it accepts 1 electron. The electron donating molecule in an oxidation-reduction reaction is called the reducing agent or reductant. The electron accepting molecule is the oxidizing agent or oxidant. A given agent, such as an iron cation existing in the ferrous state or ferric state, functions as a conjugate reductant oxidant pair, which we call the redox pair, such as an acid and corresponding base function as a conjugate acid-base pair. In redox reactions, we can write a general equation. The electron donor will give 1 electron plus an electron acceptor. In the reversible half reaction number 1, which we just saw, the ferrous ion is the electron donor and the ferric ion is the electron acceptor. Together, the ferrous ion and the ferric ion constitute a conjugate redox pair. Biological oxidations often involve dehydrogenation. The carbon in living cells exist in a range of oxidation states, as given in this figure. When a carbon atom shares an electron pair with another atom, which is typically hydrogen, carbon, sulfur, nitrogen or oxygen, the sharing is unequal in favor of the more electronegative atom. The order of increasing electronegativity is hydrogen less than carbon less than sulfur less than nitrogen less than oxygen. The more electronegative atom owns the bonding electrons it shares with another atom. With each formal loss of electrons, the carbon atom has undergone oxidation, even when no oxygen is involved, as in the conversion of an alkane to an alkene. In this case, oxidation, which is the loss of electron, is coincident with the loss of hydrogen. In biological systems, oxidation is often synonymous with dehydrogenation, and many enzymes that catalyze oxidation reactions are dehydrogenases. But not all biological oxidation-reduction reactions involve carbon. For example, in the conversion of molecular nitrogen to ammonia, 6 hydrogen ions plus 6 electrons and 1 molecule of nitrogen will give 2 molecules of ammonia. The nitrogen atoms are thus reduced. Electrons are transferred from one molecule, that is the electron donor, to another electron acceptor in one of the 4 different ways. Number 1, directly as electrons. For example, the ferrous and ferric redox pair can transfer an electron to the cuprous and cupric ion redox pair, as given in this equation. Second, as hydrogen atoms. As a hydrogen atom consists of a proton and a single electron. In this case, we can write the general equation as AH2 will give A plus 2E, that is 2 electrons plus 2 hydrogen ions, where AH2 is a hydrogen electron donor AH2 and A together constitute a conjugate redox pair, which can reduce another compound B or a redox pair by transfer of hydrogen atoms. In the reaction, AH2 plus B will give A plus BH2. And third, as a hydride ion which has 2 electrons. This occurs in the case of NAD-linked dehydrogenases. And fourthly, through direct combination with oxygen. In this case, oxygen combines with an organic reductant and is covalently incorporated in the product, as in the oxidation of a hydrocarbon to an alcohol. We have here in this equation, the hydrocarbon is the electron donor and the oxygen atom is the electron acceptor. All 4 types of electron transfer occur in cells. The neutral term reducing equivalent is commonly used to designate a single electron equivalent participating in an oxidation-reduction reaction, no matter whether this equivalent is an electron per se, a hydrogen atom, or a hydride ion, or whether the electron transfer takes place in a reaction with oxygen to yield an oxygenated product. Because biological fuel molecules are usually enzymatically dehydrogenated to lose 2 reducing equivalents at a time, and because each oxygen atom can accept 2 reducing equivalents, biochemists, by convention, regard the unit of biological oxidations as 2 reducing equivalents passing from substrate to oxygen. Reduction potentials measure affinity for electrons. Let's see how. When 2 conjugate redox pairs are together in solution, electron transfer from the electron donor of one pair to the electron acceptor of the other may proceed spontaneously. The tendency for such a reaction depends on the relative affinity of the electron acceptor of each redox pair of electrons. The readiness with which an atom or a molecule gains an electron is its reduction potential E. The tendency to lose electrons, the oxidation potential, has the same magnitude but opposite sign as the reduction potential of the reverse reaction. So reduction potentials are measured in volts, V, from an arbitrary 0 point set at the reduction potential of the following half reaction under standard conditions, which is 25 degrees Celsius and at 1 atmospheric pressure, and reactants at 1 mole. That is, 1 hydrogen ion plus 1 electron gives rise to half of an hydrogen atom. The value for E for a molecule or an atom under standard conditions is its standard reduction potential, which is denoted by E prime naught and can be determined in an experiment such as that described in this illustration. Electrons flow from the test electrode to the reference electrode or vice versa. The ultimate reference half cell is the hydrogen electrode, as shown here at pH 0. The electromotive force, the EMF, of this electrode is designated 0.00 volts. At pH 7 in the test cell, E prime naught for the hydrogen electrode is minus 0.414 volt. The direction of electron flow depends on the relative electron pressure or potential of the 2 cells. A salt bridge containing a saturated potassium chloride solution provides a path for counter-ion movement between the test cell and the reference cell. From the observed EMF and the known EMF of the reference cell, the experimenter can find the EMF of the test cell containing the redox pair. The cell that gains electrons has by convention the more positive reduction potential. A molecule or an ion with a positive E prime naught has a higher affinity for electrons than the hydrogen ion does under standard conditions. Like the values of standard free energy change, which is denoted by delta G prime naught, The standard reduction potentials may differ somewhat from those found under the conditions in a cell because the concentration of reactants in a cell are not 1 mole. In a redox reaction, electrons move spontaneously towards atoms or molecules having a more positive reduction potentials. In this type of reaction, the change in the electron potential, delta E, is the sum of the reduction and the oxidation potentials of the two half reactions. The delta E for a redox reaction is related to the change in free energy, delta G, by the following expression, which is delta G equals to minus NF delta E, where N is the number of electrons transferred. So, a redox reaction with a positive delta E value will have a negative delta G and thus will tend to proceed spontaneously from left to right. The principles of oxidation reduction energetics we just described apply to the many metabolic reactions that involve electron transfers. For example, in many organisms, the oxidation of glucose supplies energy for the production of ATP. Cellular oxidation of glucose to carbon dioxide requires specialized electron carriers. The complete oxidation of glucose, which is one glucose molecule plus 6 molecules of oxygen, it gives 6 molecules of carbon dioxide and 6 molecules of water, has a standard free energy change of minus 2840 kilojoules per mole. This is a much larger release of free energy than is required for ATP synthesis, which is about 50 to 60 kilojoules per mole. Cells convert glucose to carbon dioxide not in a single high energy releasing reaction, but in a series of controlled reactions, some of which are oxidations. The free energy released in these oxidation steps is almost of the same amount as that required for ATP synthesis from ADP with some energy to spare. Electrons removed in these oxidation steps are transferred to coenzymes specialized for carrying electrons such as NAD+, which is nicotinamide adenine dinucleotide, and FAD, which is flavin adenine dinucleotide. These are then reduced to NADH and FADH2. The reduced form of these coenzymes can transfer protons and electrons to other molecules, thereby reducing them. For example, succinate is converted to fumarate in the mitochondria as part of the citric acid cycle. Succinate loses 2 electrons and 2 protons. Now these are transferred to FAD, reducing it to FADH2. Now let's see some examples of coenzymes and proteins serving as universal electron carriers. A multitude of enzymes catalyze cellular oxidations, and these enzymes channel electrons from their hundreds of different substrates into just a few types of universal electron carriers. The reduction of these carriers in catabolic processes results in the conservation of free energy released by substrate oxidation. NAD+, NADP+, FMN, and FAD are water-soluble coenzymes that undergo reversible oxidation and reduction in many of the electron transfer reactions of metabolism. Lipid-soluble quinones such as ubiquinone and plastoquinone act as electron carriers and proton donors in the non-aqueous environment of membranes. Iron-sulfur proteins and cytochromes, which have tightly bound prosthetic groups that undergo reversible oxidation and reduction, also serve as electron carriers in many oxidation reduction reactions. Some of these proteins are water-soluble, but others are peripheral or integral membrane proteins. So that is all we have for today. Let us conclude with some key points we have covered so far. Number 1. Electron transfer in oxidation reduction reaction is an important feature in cellular metabolism since it is responsible for all work done by living organisms. Redox reactions involve loss of electrons by one molecule and gain of electrons by another. Secondly, biological oxidation reduction reactions can be described in terms of two half reactions, each with a characteristic standard reduction potential E. Thirdly, reduction potentials measure affinity for electrons and can be used to calculate free energy change of a reaction. Reactions with positive delta E have negative delta G and thus tend to proceed spontaneously. And lastly, biological redox reactions are often coupled by electron-carrying coenzymes like NAD+ and FAD.